Okay, welcome back with the next lecture of this series. This is the all about the transgenic. So transgenic, remember that to be transgenic or transfer a gene from one generation to another, it must be part of the germ line. Otherwise, it will never be transferred. If you are going to change a gene in a given tissue other than the gonads, then or other than the sperm or the egg, it will not be transferred to the next generation. So transgenic means we are going to change in the genome, and that is in the cell or in the sperm. And here is classical example with the help of the microinjection of given DNA to the nucleus and then becoming parts of that. Nowadays we can do this thing with the help of certain viruses, certain genes. They are inserted into the genome and then they are translated. And this is the classical example. Kiwis, they have produced a goat which can produce the insulin in its milk, and that is useful for the diabetic patients. The next is the knockout. Knockout means it is just opposite to the uh, cloning. In cloning, we want to express a certain gene through its genome. In knockdown, we want to not express a certain gene and then study its effect. And this whole slide it summarizes the process of the knocking down of the genome. Then another way, another technology available these days, mRNA is there, and we use the antisense DNA or antisense mRNA, and that with the help of that antisense, the given RNA in the system is basically cleaved into small part. It is never translated into a protein. So, other than knockout, another way that you keep the original copy, but you don't want that to be expressed, and that we can done with the help of the short sequences of the DNA, which will digest or which will slice the RNA component, and that is called the antisense DNA. And mostly we use certain viral vectors for this purpose. And some of the viruses vectors which are used in such studies or in such certain therapies, they are given over here. You can see the list of the viruses and then their advantages and then their disadvantages. For example, the retroviruses, they are efficient transfer, but certain disadvantages: transfers of the DNA only to the dividing cells. Insert randomly, risk of producing wild type viruses and causes immune reaction. Similarly, others, the adenoviruses, they cause immune reactions. Then but they have benefit of transfer to the non-dividing cells. The retroviruses, they can only be transferred to the dividing cells. Sorry. Uh, Similarly, the adeno-associated viruses does not cause any immune reaction. The advantage, but disadvantage, holds small amount of the DNA, hard to produce and hard to. So, the size of the fragment which you are carrying with these viruses, they are very small. Then the herpes viruses, lentivirus, and then their on. So each virus has its advantages as well as, for example, the last one is the gene gun. DNA coated in small gold particles and shot into the tissues. So does not require any vector, but its efficiency is very low. So each method, then the pressure treatment to transfer DNA. So these are basically different methods to transfer DNA to the cell to get it replicated and translated. Okay, another technique is the RFMP, restriction fragment polymorphism, and similar to that one, another technique is the AFLP, assisted fragment polymorphism. So here we use certain enzymes and we cut the DNA with the enzyme, then use certain linkers. I will explain it in the practical part, and then amplify them. And from that, after amplification, we get certain uh, markers, and those markers are used in different type of studies like the breed identification, for example, parentage identification, and many more. Again, this RFLP is useful to identify the linkage. So we have some idea about the linkage. These are the genes which are present on the same chromatid, and they transfer together. And this technique can help us to find out the linked genes. And finally, another useful technique of recombinant DNA technology is the use of DNA sequence to identify a person, that is called the DNA fingerprinting. And criminal cases, we take the specimens. In most recent, in 2018 case in Pakistan was a rape case. I hope that everybody knows about that. And they use such technology of fingerprinting to identify the criminal or the suspect. The Zainab case that was resolved with the help of this technique. So simply what you do, you got the DNA and then you run on the gel, and then you have certain probes, and those probes are attached. So now you have one DNA evidence. Evidence means that is the DNA which is where the samples or specimen which you have collected from the crime scene, and suspect you have many individuals who are suspected, and you get their DNA and then you run them and then probe them, and after probing you get the results. And here in this case you can see the evidence and suspect, they are 100% matching. The other individuals, the suspect 2 or the other individuals, they have different type of bending patterns. The only one, the suspect 1, that is having 100% same bending pattern as that of the evidence or the DNA collected from the crime scene. So this is another technique which is very useful, and it is used in the fingerprinting, especially in criminal cases, to resolve them. Thank you very much, and that's all about the part 3. Uh, if you have any question, please raise on the channel or on the Google class or on the UAAR class, Arid Agriculture University classroom. You can ask the questions and I will try to resolve them. And with the end of this chapter or this part 6, we have completed our final term course, and in this way the entire course of ABG 202 or ABG 1, Animal Breeding and Genetics 1 course of Doctor of Veterinary Medicine is completed. The theory component is completed. The practical component, I will give you some actual real-time lab work videos and a couple of videos, PPT videos in future. And please prepare for your course, for your exam very well. Thank you very much. Have a good time.